In these pictures, we are doing an experiment where Gloriana and Cecilia are dropping the blood on the paper's surface while Grace is measuring the height from where the blood is dropped. This experiment is being done to see how height of where the blood is dropped affects the diameter or pattern of the blood drop. Have you ever wondered why you can't mix different blood types when you're getting a blood transfusion? Demonstrated in this, you can see that there are different types of blood cells and their corresponding names. All of the blood types are different because of antigens and antibodies. Antigens are the little lines sticking out of the blood cells, but in reality, they are little types of structures that can cause an immune response to take place. Antibodies, on the other hand, are a type of protein that circulates in your blood and interacts with a certain antigen by producing a response to counteract a particular antigen that is introduced when a different blood type is entering your body. Antibodies, on the other hand, are a type of protein that circulates in your blood and interacts with a certain antigen by producing a response to counteract a particular antigen that is introduced when a different blood type is entering your body. An antibody connects to an antigen to mark them so another cell can get rid of the blood cells that are a different blood type. This action causes a reaction that leads blood to clot and which will kill the person that's receiving the wrong blood type. DNA is found in the nucleus organelle and is also found in the cells of all living things. DNA's code provides the instructions to build proteins. DNA lays the foundation for how organisms grow, what they look like, and their ability to get diseases. DNA is inherited. One of the DNA's functions is to tell amino acids how to line up and form themselves into protein shapes. DNA is made up of nucleotides. Nucleotides are formed by the combination of a phosphate group, a sugar, and 4 different nitrogenous bases: adenine, thymine, guanine, and cytosine. Adenine always pairs with thymine, and guanine always pairs with cytosine. When DNA is turning into messenger RNAs, thymine is turned into uracil, but all the other pairings stay the same. DNA makes the antigens that are outside of the top layer of blood. These antigens are the ones that show what blood type you have. Inside the nucleus is where DNA is located. There is a process called transcription. Transcription is the process where the DNA code sequence is being copied to make a messenger RNA. RNAs are chemicals inside the nucleus that make partial copies of the DNA code. RNAs are shorter and missing one of their sides, and they're small enough to fit through the nucleus, out the cytoplasm, and into the ribosome. But to be able to do that, you need a special enzyme that separates the 2 DNA strands so that the code can get out of the nucleus and into the ribosome. And that enzyme is helicase. Helicase breaks through the hydrogen bonds that hold the DNA bases together. After that, the polymerase enzyme replicates DNA molecules to build a new strand of DNA that matches the previous one. In this experiment, we took blood from the suspect's arm to compare it to the blood found in Anna Garcia's crime scene.